Welcome, class, to week 8 of plant physiology. Tonight we'll be looking at lecture 9, the phloem. So we've already started looking at the vascular tissue, the all-important xylem. And of course, we will now focus on phloem function and structure, and then really take a closer look at how carbohydrates are transported through the phloem. And that process is called translocation. All right, so let's run quickly through these basics here. Survival on land poses serious challenges to plants. Obviously, they need to acquire and retain water, and they also need systems designed for long-distance transport of the products of photosynthesis. We've looked at the xylem, of course, which transports water and minerals from roots to aerial portions of the plant. And tonight we're going to focus on the phloem. And the phloem, of course, moves or translocates products of photosynthesis from mature leaves, or mostly mature leaves anyway, but from any part of the plant that can photosynthesize, to areas of growth and storage, including the roots. So the word translocation simply refers to movement of the products of photosynthesis, that is photosynthates, from a source where they're synthesized to a sink, to part of the plant that's called a sink. And of course, this translocation occurs in the phloem. So that term photosynthates obviously refers to the carbohydrate products of photosynthesis, and also modified carbohydrates that are synthesized after that. The phloem also transmits signals between sources and sinks, and redistributes water and compounds. We know that the two transport pathways found within the vascular tissue, that is the phloem and the xylem, extend right throughout the plant body. The phloem is generally found on the outer side of both primary and secondary vascular tissues. And we've got an example of each on this slide. And you've probably already seen these as part of your DIY kit assessment. So you've probably seen exactly what we're looking at here. So the illustration, the photo on the left, is showing primary phloem. So this is a transverse or cross-section of a stem of clover. And it's showing the primary phloem, labelled as such here, to the outside of the primary xylem. Then the photo on the right shows a 3-year-old stem, also a transverse section here, and this is a stem of an ash. And you can clearly see that there's secondary growth occurring, obvious by 1, 2, 3 growth rings. And you might recall that growth rings, of course, are formed by seasonal growth of secondary xylem, with this layer of secondary xylem here being the most recently formed. So of course we've got the vascular cambium here, and to the outside of that we've got the most recently formed secondary phloem. The older secondary phloem and the primary phloem have long ago been crushed as the stem expands through the development of the secondary xylem. All right, so let's take a closer look at the structure of the phloem. So the cells, the main cells of the phloem, are called sieve elements. And this includes sieve tube elements that you would find most commonly in the angiosperms, and sieve cells that you'll find in gymnosperms. We'll be focusing, of course, on the sieve tube elements tonight. But in addition, phloem tissue itself not only includes sieve tube elements, but also companion cells and parenchyma cells. The parenchyma cells are the ones that store and release our food molecules. And in some cases, phloem tissue also contains fibres, cells called fibres, and other cells called sclerides, and these function in protecting and strengthening the tissue. But it's only the sieve tube elements that are directly involved in the translocation of photosynthates. The other cells within phloem tissue are merely support, storage, transport cells. Okay, move on from there. All right, so let's look at sieve tube elements, or simply sieve elements. Mature sieve elements are quite unique among living plant cells in that they have the most basic components within the cell. They're so highly specialised. As they're actually developing and differentiating into sieve elements, they lose their nuclei, they lose their tonoplasts, that is their vacuolar membranes. They lose many components of the cytoskeleton, that is microfilaments and microtubules, and Golgi and ribosomes also. They are living plant cells though, so they do retain the plasma membrane. They do have a small number of modified mitochondria, some plastids, and some endoplasmic reticulum. They're different to the tracheary elements of the xylem, which, as you probably remember, lack a plasma membrane. They have lignified secondary cell walls, and they're dead at maturity, at functional maturity. So remember, the sieve elements are actually living cells. They're not generally lignified cells, although they often undergo secondary thickening of the cell walls. Which probably suggests something to you about the type of pressure that exists within the phloem sieve cells, in contrast to the xylem cells. What do you think that difference would be? So you remember we had that negative pressure that forms in the xylem, and hence that need for the lignin strengthening of the cell walls. But given that phloem don't tend to be lignified, do you think that there would be negative pressure in the phloem? Positive, absolutely. Less pressure, and it's positive pressure. So we don't have that requirement here in the phloem to have those super strong cell walls. Absolutely right. All right, here we have a nice illustration of a mature sieve element. On the left, just one sieve element illustrated here. So the main components that we can see is the end walls of this one cell, and they're called sieve plates, and they contain these large pores which allow movement, continuous movement of photosynthates from one sieve element to the next. And then, of course, in the cell wall, they have these areas of tiny pores called lateral sieve areas. So obviously this allows a connection, a lateral connection between adjacent sieve tube elements. Okay, in B we have two sieve elements joined end to end. And here we can see some of the components that are labelled here. We've got some plastids, a little bit of smooth endoplasmic reticulum. The cytosol is intact, the plasma membrane is intact. We've got a thickened primary cell wall, and adjacent to this particular sieve element here, we have another component of the phloem, that is one type of companion cell, which is also always very closely associated with these cells. So you can imagine, because these sieve elements are lacking in a lot of the normal organelles and structures that you'd find in living cells, that the companion cell actually takes over many of those metabolic functions that are required to sustain the cell. And this is just an ordinary type of companion cell here. There's three major different types of companion cells, and we'll visit them briefly in a few minutes. All right, let's recap. So sieve elements have characteristic sieve areas in their cell walls where those small pores interconnect adjacent sieve element cells. The sieve plates that we find on either end of the cells have larger pores, and they're found on the end walls. Sieve plate pores are simply open channels. They're not covered by any type of membrane, and these allow transport between each of the sieve element cells. Sieve element sap. So what does it contain? It's obviously rich in sugars, and there's other molecules also, which we'll look at in just a minute. A loss of sap due to damage has to be prevented. So any sort of wound, any sort of damage to the phloem requires some sort of mechanism to repair or prevent any further loss of the phloem sap. Two major mechanisms which are described: a short-term sealing mechanism, and this involves a certain type, or a group of proteins, similar proteins called sap proteins, and they're referred to as P proteins. And it's thought that these P proteins, which, by the way, are synthesized by the companion cells, and they're found in a very high concentration in the sieve elements, and they appear to function by actually plugging the sieve pores in the sieve plate to prevent any loss of sap. Then we have a long-term mechanism which involves the synthesis of callose. Callose is a type of polysaccharide, a modified type of polysaccharide, and the callose is very quickly synthesized by the sieve elements themselves in response to damage and in response to other stresses also. Heat stress, high temperatures, for example, can kick off the synthesis of callose, and the callose will basically also block the pores of the sieve element. So these images here, we can see on the left, this is an open sieve plate here, which hasn't been sealed in any way. And then on the right, we can see this plug here, almost like a fluorescent area here, and this represents a plug of callose. And then what happens when the sieve element actually recovers from the damage or from the stress that it's under? The callose is simply dissolved by a callose dissolving enzyme, and those pores can start to function once again. All right, here, this is just an example of the plant's response to pest attack. This is showing callose deposition being induced in rice plants that are being attacked by plant hoppers, brown plant hoppers. And it's interesting that this response occurs in both resistant rice plants and susceptible rice plants. So where the difference lies is that in susceptible plants, the feeding by these insects also activates genes for a callose hydrolyzing enzyme. So that is the insects feeding, and there's some sort of signal which triggers that callose enzyme, the enzyme that breaks down the callose, which enables the insects to feed even further, because it keeps that phloem sap running through the actual phloem. So this allows the insects just to feed continuously, and it results in decreased sucrose and starch levels in the leaf. So that's how some plants are susceptible, and obviously this implies that in resistant plants, that callose hydrolyzing enzyme is not activated. So the aphids, the plant hoppers rather, tend not to get very much out of feeding on the resistant plants, because the sieve element has been blocked. All right, so as we saw earlier, each of the sieve tube elements is associated with one or more companion cells. These are supportive cells. And it's interesting that division of a single mother cell results in the formation of one sieve tube element and the formation of one companion cell. The companion cells function in the transport of photosynthates from producing cells in mature leaves to the sieve tube elements in the minor veins of the leaf. The companion cells also take over some of the critical metabolic functions that are reduced or lost in sieve tube cells, as we said earlier. So, for example, it's been shown that companion cells supply most of the ATP required by those sieve elements. Here are the three different types of companion cells that are found. Ordinary companion cells, which have a cell wall with just a smooth inner surface. The second type are the transfer cells, and these are companion cells that develop these ingrowths of the cell wall, primarily on the walls that face away from the sieve element, and these ingrowths greatly increase the surface area of the plasma membrane, which results in increased potential for solute transfer. Transfer cells have very few plasmodesmata, so they're more suited to apoplastic transport. Now, in contrast to the transfer cells, intermediary cells have lots of plasmodesmata connections to surrounding cells. So these are better suited to symplastic transport of sugars from the photosynthetic mesophyll cells to the sieve cells. And that's illustrated here for you. So in this image in A, we have the ordinary companion cell here, which is just the smooth inner cell wall. Now we can compare that cell there to this one. This is a transfer cell, and we can see these ingrowths of the cell wall in several sections. And of course, the ingrowth of the cell wall means that we have an ingrowth also of the plasma membrane, or rather an increased surface area of the plasma membrane. And down here we have the intermediary cell. And we can see lots of plasmodesmata here, indicated here by the black arrows. So a whole region of plasmodesmata here and also over there. And here's a section of plasmodesmata here. All right, so now let's talk about patterns of translocation. So translocation, or movement in the phloem, doesn't occur exclusively in an upwards or downwards direction, and it's also not controlled or defined by gravity. Rather, it tends to move from an area of supply, called the source, to any area of metabolism or storage, which is called a sink. So let's think about what part of the plant would act as a source. So obviously the major source in any plant would be the mature leaves. But there's also sources that would probably surprise most people depending on the plant species. So sources will include exporting organs, of course mature leaves, which we just said, and we're talking about mature leaves here as sources because they are the leaves that can produce photosynthate in excess of their own needs. So they've got basically leftover that they can export. When we talk about young leaves, before they're actually fully developed, they tend to act as sinks. And then once they're completely developed, or even as young leaves, then they'll start photosynthesizing enough to start exporting. But let's think about other sources that you might expect to find in a plant. Can anyone give me any ideas? Oh, that's all right, internet issues. As long as I don't have them and keep dropping out, which happened one night. Oh, sorry, I missed that question. Are the resistant plants GE? GE, resistant plants GE. Sorry, what does that mean?
You can hear me now. It's plants that are resistant to the insect attacks.
Oh, yes, yes.
Are they genetically engineered to be like that? As far,
No, they're not genetically engineered at all. And even though they say that they're resistant, I don't think that's really an accurate term. We like to use the term now. We prefer now to use the term tolerant rather than resistant. Yeah, because really there's no plant that is completely 100% resistant.
Okay.
Yeah, so more tolerant. But no, they're not genetically engineered at all. No, this is all comes about through traditional plant breeding.
Yeah, for sure.
Yeah.
Phylloxera resistance, rootstocks and things like that.
Yes. Yeah. Oh, that's right. You do. You have resistant rootstocks to phylloxera. Thank you very much. Yes.
Okay.
Yeah, good question. All right, so we're talking about sources. What part of a plant would act as a source? Do you think can act as a source? So all we've got here so far is mature leaves. Any idea? Senescing, yeah, actually that's a good one. Senescing leaves can actually, well, they do act as a source, yes, before they completely drop off, yep. in general, what parts of any plant could act as a source, just in general? Lignifying starchy bits. Yeah, starchy bits can actually switch from being a sink to being a source. Yep, absolutely. Yeah, what I was actually after was that any green part of a plant can potentially act as a source. Any green part of the plant. Yeah, exactly right. Green bits, absolutely. So the stem, with wheat, you might not be familiar with the structure of the wheat, the ear, the head, where the grains are developing, but they've got these outer layers surrounding the inflorescences called glooms and lemmas. They're just like these protective sepal-like structures. And even though they're so small, they're incredibly strong sources. Quite amazing to think that such a small part of the actual developing inflorescence is such an important source of photosynthase. So there are our sources. And here we have a great example of a source that can become a sink. So as someone said a little bit earlier, we can think about storage organs exporting its stored sugars. Oh, sorry, I'm confusing myself here. Let's just go to this part here. Example, storage root of wild beet. Okay, it acts as a sink, obviously during the growing season, so it's importing lots of carbohydrates, and then during the second season it actually becomes a source, so it starts exporting those carbohydrates to start producing a shoot, which will then produce the reproductive structures. So this is a really good example of a sink that then becomes a source. And I think that goes back to what you were saying, Lachlan, about the senescing leaves. The same sort of principle applies here. All right, what about sinks? What would be, it's all listed here for you. All right, so sinks obviously include any non-photosynthetic organ and organs that don't produce enough photosynthase to export. So the classic sink tissue, of course, are the roots. They don't photosynthesize at all, so they need to import all the photosynthase that they require. Then we've got tubers. Developing fruits are very strong sinks, and immature leaves, as we said earlier. All right, so we've said that photosynthase will be translocated from source to sink, but the overall pattern of transport is a lot more complicated than simply source to sink movement. It depends on a lot of factors, and some of these factors are still not well understood, and that's why they're constantly looking at different ways to try to determine, what directs the allocation of photosynthase to one part of the plant at a certain rate, and then what changes that rate to redirect the photosynthase elsewhere. But the pattern of transport depends on proximity of the actual source to the sink, the developmental stage, the vascular connections, modification of translocation pathways, and they do know, they've done some nice experiments to show that certain sources mostly supply specific sinks. So I don't know about grapevine, you can probably tell me, but I'll tell you about wheat. They found that the upper leaf, which is called the first flag leaf, and then the second leaf down, which they call the second flag leaf, they tend to be the strongest sources that supply the developing head of grain. They're not the only two strong sources, but they're certainly two of the strongest. So what do you think about grapevines? What do you think would be the strongest source that's supplying the developing berries, for example? And you call that an inflorescence, do you? Is that called an inflorescence? The leaf directly across the bunch. So the closest leaf, is that's thought to be the strongest source?
Yeah, generally the leaf across the stem from the bunch is
right.
seen as the strongest source.
Right. Okay.
And usually the one that we take PDL samples from as well.
Yes. Yeah.
Yeah. So
okay.
It's usually seen as they're the main powerhouses for the bunch, apparently. And then if you take those leaves off, then it goes up to the next leaf nearby.
Oh, right. Okay. Yeah. So very similar to wheat then.
Yeah.
Very similar to wheat. Yeah.
The most direct path.
Okay. And yeah, so you've got obviously on a grapevine, you've got several bunches of fruit on the one stem.
Yeah, 2 or 3 usually. Yeah,
2 or 3 bunches per stem, and then would it be the leaf immediately before the bunch,
or across, like the bunch
or
is
across?
On one side
Okay.
of the stem, and then the leaf is directly across on the other side of the stem.
Oh, right. Okay. Yeah, I'm with you.
The one that's I'm pretty sure I've read or heard that. It's one directly across.
Yeah. Yeah. No, it's very interesting. I'll just draw something. You might have seen this in the textbook, which I find quite interesting. And I'm sure they've done these experiments in grapevines and lots of different species. But what they did, they radioactively labelled a leaf. So this was the leaf that they labelled with I think it was carbon-14. So radioactive carbon. And then they could trace the movement of photosynthetes to different leaves. And I can't draw this very well at all. But what they found, and I'm doing a very bad job at drawing it, but what they found was that the carbon-14 from this leaf was translocated to new developing leaves on one side, just on one side. So nothing was going here and nothing was going here, even though these new leaves on the other side were at the exact developmental stage of these leaves. So I found that quite interesting, that it's not only proximity, but also the side of the plant that leaf is appearing on, if that makes sense. So I wonder, and of course I'm speaking as a complete novice here, if you've got a grape bunch developing out here, so that's your bunch, if that's your bunch, and you've got a leaf here, and I can't draw a grape leaf, but you can, there you go, say that's the leaf. I'm wondering if perhaps that leaf might be supplying this bunch up here rather than this bunch down here.
Yeah,
if that makes sense. Yeah, I don't know. I'm just surmising here.
I think it was an AWRI thing that I was. They were talking about leaf plucking.
And,
and people take those leaves off,
right?
Bunches to try and get some leaf area, or to get some airflow going.
Oh yes,
People were asking a question of taking those leaves off around the bunches, whether they're going to be restricting photosynthesis to the bunches.
Yeah.
And they did say.
And do you know what they found?
Well, I
Do you
think
know?
they've done the same thing with carbon-14 in those leaves. And
Okay.
they found that leaf directly across from the bunch is the powerhouse.
Oh, right. Okay, so it works differently depending on the plant that you're looking at.
Yeah, I
Yeah.
think so, but I'm pretty. This was 3 or 4 years ago that
Okay. Yeah.
I'd heard that.
Mm.
Um, so yeah, and I think if you take that direct leaf off, then it goes up to the next leaf or the next
Yeah.
leaf sort of thing.
Yeah. No, it's very interesting. There's certainly a lot that we still have to learn about translocation.
That's good.
Yeah, no, it's great. That's great. Okay, I think that's it on this slide. Let me just have a quick look. All right, we've covered all that. YouTube video. I still haven't watched that YouTube video, but I will get to it. All right, so let's look at materials translocated in the phloem. Obviously, water is the most abundant substance in the phloem, and there's lots of carbohydrates, predominantly carbohydrates, but there are also amino acids in the phloem sap, hormones, some inorganic ions, RNA molecules, and some proteins, and also some secondary compounds involved in defense and protection. Carbohydrates are the most significant substances in the phloem, with sucrose concentrations of 0.3 to 0.9 molar. And of course, even though we always talk about sucrose because it's present in the highest amount in the phloem, there are an incredible number of different types of sugars that are transported in the phloem. I must find a study that was done years ago, and there were lots of different ones. They were present in minute quantities, but it was amazing to see all these different types of sugars that were present. Anyway, here we go. Table 10.2, composition of phloem sap from castor bean. Sugars present in by far the highest concentration, then amino acids, organic acids, a little bit of protein, bit of potassium, chloride, phosphate, and magnesium. So that gives us a really good overview there. All right, reducing sugars. Who remembers reducing sugars from living systems? And of course, they're listed here, well, the common ones here: glucose, mannose, fructose. Not really. Now you're really making me feel bad. You don't remember it. All right, glucose, mannose, fructose are not translocated in the phloem because they are quite reactive. So I best not ask you why we call them reducing sugars. Okay, I will move on. No answer. Please don't. All right, it doesn't matter. It really doesn't matter. You don't need to know. But you might remember, I might jog your memory, that when we talk about these monosaccharides, if you remember a little bit of your organic chemistry, you might recall that they contain aldehyde groups or ketone group. And that's the part of the monosaccharide that can be reduced or oxidized. And I know you remember it, but like I said, it doesn't matter. We will move on. The important point on this slide is that it's not those reducing sugars that are translocated in the phloem. Instead, non-reducing sugars are commonly translocated, and also the two sugar alcohols, mannitol and sorbitol. So we know that the most common non-reducing sugar that's translocated is sucrose. And we have one of the sugar alcohols shown here. This is mannitol. And this is the product of the reduction of mannose, which forms mannitol. Okay, so also what's shown here is sucrose, of course, which is 1 fructose and 1 glucose. But then if we combine sucrose with 1 galactose, that gives us the non-reducing sugar raffinose, which is also translocated in the phloem. If we join 2 galactose monosaccharides to sucrose, that gives us stachyose, and 3 galactose monomers bound to sucrose gives us verbascose, which is also another non-reducing sugar. All right, and that's basically the take home from this slide. So how do they analyse phloem sap? Obviously very challenging to isolate pure phloem sap that hasn't been modified in some way due to the method used to extract it or contaminated with contents from other cells. And you might remember that this is the most preferable approach, to use a feeding aphid, which inserts its mouth part, its stylet, into the cells and directly into the phloem. So this is the stylet here being inserted into the sieve element. And then, of course, what they do, they use a laser to cut off the stylet, and then they can actually collect the phloem sap from there. I mean, there are problems also associated with this method, but it's the best method they've got at the moment. And that's the whole methods described for you here. All right, let's look at other solutes in the phloem. Nitrogen is translocated in the phloem, and it's found in the form of the amino acids glutamate and aspartate. The plant hormones translocated include auxin, gibberellin, cytokinins, and ABA, abscisic acid, and we'll come back to the important plant hormones in a later lecture. There are low levels of proteins found in phloem sap, low levels of RNA molecules. There are some messenger RNA molecules and also some small regulatory RNA molecules. And as we saw earlier in the table, some inorganic solutes, potassium, magnesium, phosphate and chloride, but only small amounts. These solutes here are immobile in the phloem. You never find them in the phloem. Well, they haven't so far. Nitrate, calcium, sulfur and iron. All right, now let's get on to the important mechanism which is thought to best describe how photosynthates are transported in the phloem. What is the mechanism that drives translocation? And I'm sure you've heard of this, the pressure flow model of translocation in the phloem. This model proposes that translocation occurs as a flow of solution by mass flow or bulk flow, and this is driven by differences in pressure. So a pressure gradient that exists between the source and a particular sink. Who remembers this model, the pressure flow model? Because we just touch on it briefly in living systems. But you can tell me that you've forgotten. I won't feel too bad. Remember the name only. Okay. At least you're honest. At least you're honest. All right. The pressure flow model is it's based on a passive mechanism for phloem transport. It was proposed in 1930, and it basically involves phloem loading at the source, so loading of photosynthates into the phloem at the source, and then phloem unloading, so unloading of the photosynthates at the sink, and this is thought to establish a solute gradient, and as a result of that solute gradient, this establishes a pressure gradient, and it's the pressure gradient that actually drives translocation. So let's think. Oh, someone's dropped out. So let's consider diffusion, the process of diffusion. They know that the mechanism of diffusion is far too slow to account for translocation in the phloem. Far too slow. They know how fast, for example, a molecule of glucose takes to diffuse from one side of a cell to the other. It's something like 2.5 seconds for one glucose to diffuse across a cell, which has a diameter of, say, 50 microns. So they can easily calculate how long it would take for a substance to diffuse over 1 metre, a distance of 1 metre. So if translocation was purely based on diffusion, then translocation would take 32 years just to travel a distance of 1 metre. So it's clearly not diffusion. Clearly there's something else going on here, and hence the pressure flow model that was proposed. Right, the pressure flow model proposes that there are 3 different mechanisms required to generate high concentrations of sugars in the sieve element at the source end. 1, photosynthetic metabolism in the mesophyll cells, obviously the source of production of the photosynthates. Then conversion of the photosynthates to transport sugars in one type of companion cell called the intermediary cells. I refer to this as polymer trapping. Don't worry about that too much. And the third mechanism is active membrane transport. And we'll come back to these mechanisms in just a little bit. But first, let's look at how the pressure flow model is thought to work. So we'll just go through this explanation and then we'll look at this on the next slide. So this slide and the next one, very important slides to revise going into the final exam. So in source tissues, accumulation of sugars occurs in the sieve elements. In other words, the sugars are loaded into the sieve element at the source, and this generates a low solute potential, that is a more negative solute potential. This low solute potential causes a decrease in the water potential. In response to this water potential, this lowering of the water potential, water will move from the xylem, from the adjacent xylem, into the sieve element. This causes an increase in turgor pressure in the sieve element. At the other end of the translocation pathway, that is at the sink end, phloem unloading occurs. So the photosynthates are moving out of the phloem into the cells, the sink cells that need the carbohydrates. This leads to a lower solute concentration in the sieve elements at the sink end, which generates a higher or more positive solute potential in these sieve elements of the sink tissues. As the water potential of the phloem rises above that of the xylem, water will then move out of the phloem in response to that water potential gradient. That water will move out of the phloem, back into the xylem, and this results in a decrease in turgor pressure at the sink end. That sounds quite complicated looking at it on this slide. Let's try to simplify it by looking at the image. All right, so let's start with our source cell up here. There's our source. So this might be a leaf cell, a mesophyll cell, for example, and here's our sink cell, which might be one of the cells in the roots, for example. But keep in mind, even though we're going to look at translocation in a downwards direction, obviously this occurs just as easily in an upwards direction. All right, so the sugar at the source, illustrated here by sucrose, which are the red spheres, is actively loaded into the companion cell and then sieve cell, adjacent sieve cell. And for the purpose of translocation, the companion cell and the sieve cell are considered to be one functional unit. All right, so we've got loading, phloem loading here at the source. So what does this do to the solute potential? What do you think that does to the solute potential or osmotic potential? When we're increasing the concentration of sucrose. Correct. It decreases or lowers this figure here. Yes, it becomes more negative. Correct. If this becomes more negative, that is the solute potential, then our water potential also becomes more negative, doesn't it? Yes. So we've got a water potential here of negative 1.1 megapascals in the phloem. Let's have a look over here at the water potential in the xylem. This is negative 0.8 megapascals. This is higher here, obviously, than it is here. So water will move from the xylem into the phloem. Absolutely, we'll load up with water. We'll load this up with water, and that results in an increase in turgor pressure. So we've built up the solute concentration. That results in an increase in turgor pressure because the water obviously follows. And this is why the phloem elements and the xylem elements are always packed closely together. You never find them separate from each other. They always exist wherever you look in the plant as a bundle, as a vascular bundle, because they're working together in this way. All right, so we've built up the pressure here. Now let's look at what happens. Now we have pressure-driven bulk flow or mass flow of water and solutes from source to sink. Okay, why is it going from here to here? Okay, and the answer's just been given to you. Why is it moving from that particular sieve element there to this one down here? So look at the numbers. Look at the numbers and explain to me, by looking at the numbers, what is driving that movement? What's driving that movement? Turgor pressure, exactly. So look at the pressure. So we've got the pressure here, turgor pressure. This is a positive number, positive pressure, positive number, unlike the negative hydrostatic pressure in the xylem, which also always has a negative value. 0.6 megapascals here compared to 0.3, lower pressure here, higher pressure here. This is what's supposed to drive translocation from source to sink, the actual pressure gradient, the difference in pressure. That's why it's referred to as pressure-driven bulk flow. If you were to actually compare the water potentials, that would be very confusing and it wouldn't make sense. The water potential up here, negative 1.1 megapascals. Water potential here, negative 0.4 megapascals. That would not make sense for translocation to occur in this direction based on water potential values. But it does make sense if we look at the pressure potential values. Okay, so once we've moved water and the carbohydrates, the sucrose, down to the sink. What happens here? Unloading. The sucrose is unloaded at the sink. Here's the molecules. Need the pressure to wash down. Yeah, essentially that's all it is. The actual pressure is moving the photosynthates, or the water, even against a water potential gradient, because it's simply this pressure-driven mass flow. More solutes. Oh, sorry, I'm missing what you're saying because I've got this in too much. That's better. Okay, because more solutes would normally draw water instead. Yeah, absolutely right. Correct. You've got more solutes up here, drawing water out of the xylem. Drawing the water out of the xylem increases the pressure, and that pressure will push the phloem sap from high pressure down to a low pressure, or up to a low pressure area, up or down. Good. All right. Complicated, but you've obviously nailed it. Good. All right, phloem unloading occurs here, unloading of sucrose into the sink cells. So we're just saying that these are root cells, for example, but they could equally be your fruit, your developing fruit, or the developing ear or head of wheat, which obviously is moving. That means that translocation's moving in an upwards direction. All right, so as the unloading occurs here, phloem unloading, what does that do? It increases the solute potential. Here we go, solute potential, negative 0.7 megapascals. Let's compare that to what we have in the adjacent xylem, which is shown as being separated, physically separated, but it's not. They're butted right up against each other. Here's the solute potential, negative 0.1 megapascals. So it's higher here, it's lower here. And we can look also at the water potential now, negative 0.4 versus negative 0.6. So we've got a higher water potential here, lower water potential in the xylem. So water will actually move out of the phloem back into the xylem. So this basically describes the movement, translocation in the phloem. This would be a very good slide to study, and of course the description also on the previous slide. But I think the descriptions here are sufficient, really, to explain the pressure flow model. All right, so you're both happy with that? Excellent. All right, good question. Is that a fair representation of numbers? Yes, it is, but there would be a difference, definitely depending on plant species and depending on growth, depending on temperature, nutrients, photosynthetic rate, light intensity. There would be a lot of factors that would affect what's going on here in with photosynthetic movement. Normally the same length. Yeah, awfully convenient. Xylem and phloem tubes normally the same length. Yes, they are. They are. It is an amazing, amazing piece of engineering of nature. It's incredible. And they're working together all the time, everywhere you look in the plant, whether it's in the stem, whether it's in the roots, in the leaves, you know, right through into the reproductive tissues. So if you look at the stamen with the anther, that vascular bundle comes right up right up to where the pollen's developing, that central vascular bundle, and there they are, phloem, xylem packed tightly together. Balanced out somewhere. Maximum distance photosynthate can move. You mean laterally? Can it move laterally through the cells? Is that what you mean? So if you've got in a tall tree. That's a good question. Photosynthetes. So you're saying if photosynthetes produced in one leaf, how far can it actually move through the actual plant? That's a good question. I can't answer that. I don't know what the maximum distance would be. I'd say it would be substantial, because if you picture a tall tree where the lowest leaves are meters above the ground, many meters above the ground, then you can imagine that photosynthetes have to be transported from those leaves all the way down to the roots. Yeah, good question. Good question. I don't know if they've done those studies, but I'm sure there's still a lot of unanswered questions. Could photosynthate go from the top of a tall tree all the way down to the roots? Yeah, I don't know. Don't know. Good question. I know it could definitely travel a fair distance.
The reason I'm asking is because I wonder if, say, if you had something like a mountain ash or something like that, that's a super, super tall tree.
Yes.
And the photosynthetes have to go from the leaves, which it might be 100 meters or whatever it is.
Yeah, well, the tree itself is about 100 meters tall.
That's right. So
even greater than 100 meters, yeah.
Most of the source is 100 meters above the roots.
Yeah.
As the tree is growing, the photosynthetes actually just build up in the tree, and they just, it's hard to know how far they can actually travel through a tree.
Yeah.
Whether it's just like a block pressure sort of thing where the photosynthetes that are hitting the roots at any one time might have been produced many, many, many years ago, possibly, or something.
Yeah, I don't know. I think maybe Lachlan's onto something here where he's saying multiple pressure gradients rather than one. Tallest regnans, about 96, 130, eucalypts. I think isn't the ash over 100 metres? Oh, 100, yeah. Look, I imagine that there would be different pressure gradients in different sieve tube elements. So you know how you've got multiple sieve tube elements within an actual bundle itself. As far as I know, each of those sieve tube elements can act independently of the one next to it. So in my mind, that means that you could have one sieve tube element. So just think of one tube that's extending right through the tree. One tube could be transporting in one direction. The one next to it could be transporting in another direction. And then, of course, where those photosynthetes are unloaded will depend on the sinks that it meets along the way, if that makes sense. So you could have one that's going upwards that basically unloads before it even gets to the top. And the one that's going down might be able to go all the way down to the roots, because that might be the strongest pressure gradient that's actually present at that time, at that particular point in time. They found one cut down but none left standing over the ton. Oh, is that right? Are you talking about the eucalypts, Lachlan? Okay. Oh, that's interesting. I thought they were over 100, but you're saying there's none left. Yeah. Oh, the redwoods, Californian redwoods. Yes, they're taller. They're the tallest as far as I know. The redwoods. Yeah, look, it's really interesting. Yeah, that's what I thought. Regnans is the tallest. Yeah, no, very interesting. All right, we should move on. As fascinating as this is, and wouldn't you love to get a radioactive tracer into your favourite grapevine and work out exactly what's going on with translocation, particularly under different conditions and different stress conditions, you know, different conditions of pathogen attack or pest attack. It would be really fascinating to see. All right, so still on the pressure flow model. Now, if there were no cross walls present in the sieve tube elements, that is, if that tube, that pathway was just a single compartment, then the different pressures at source and sink would equilibrate. So those cross walls, those sieve plates with the pores, are essential. They present a sequence of resistances to the moving phloem sap, and this resistance results in generating and maintaining a pressure gradient between the source and the sink. Here we go. Mass flow permits the flow from a source organ with a lower water potential to a sink organ with a higher water potential. Let me just go back. This is what I was referring to. Show previous page. Low water potential to higher water potential. Here we go. So negative 1.1 megapascals versus negative 0.4. Yep. So from lower water potential to higher water potential. And that doesn't make sense, does it? Is this a violation of the law of thermodynamics? No, because we're talking about mass flow driven by pressure gradients, not simple osmosis. Oh, Luke, you would like to study with radioactive tracers. You might have to organise that somewhere for your third year research project. Would be fantastic. Okay. Chapter 1, Chapter 5. Right. So in summary, movement in the translocation pathway is driven by transport of solutes and water into source sieve elements and transport into source sieve elements and out of sink sieve elements. Oh, yes. Okay. Now I am confusing myself. Yes, that does make sense. Transport of solutes and water into the sieve elements at the source and out of sieve elements at the sink end of the pathway. This is a passive, pressure-driven, long-distance transport, which depends on phloem loading and unloading, as we said earlier. And these mechanisms are responsible for setting up that pressure gradient, as we saw. Okay, terrific. All right, let's just look at what I think is a terrific little experiment that's been done. Well, I shouldn't say a little experiment. This would be technically quite challenging to take an intact leaf from an intact plant and slice a little window here, without actually damaging the phloem, and another little window here. Then placing the objective from a confocal scanning laser microscope over this window, looking into the phloem, and then adding a dye, a fluorescent dye, into the phloem here, somehow without damaging it, and then looking at whether that fluorescent dye appears in this window as you're looking down through the microscope. And which of course it did. So this shows that the direction of translocation in this leaf, and I can't remember what plant they used, was in this direction. Translocation was occurring towards the intact plant. And this is what they actually saw. It was a green fluorescing dye that was added. And these are the sieve elements here. So quite an interesting little experiment. Technology is amazing, isn't it? But more amazing are the experiments that people actually come up with. Fantastic. Let me just clear my my drawings. Don't want to keep seeing them. Clear. Better. All right, several steps are involved in moving photosynthates from the mesophyll cell chloroplasts to the sieve elements in leaves. Let's look at these steps. Step number 1, triose phosphates from photosynthesis are transported from the chloroplast, out of the chloroplast, into the cytosol, and there they are converted to sucrose. Other transport sugars are later synthesized from sucrose. Step number 2, sucrose moves from the photosynthetic mesophyll cells to cells near the sieve elements in the smallest leaf veins, and this is referred to as short distance transport. And this short distance transport pathway usually covers a distance of only a few cells. Step number 3, phloem loading. Sugars are transported into the sieve tube elements and companion cells, or rather the other way around, into the companion cells and then into the sieve elements. Sucrose is then translocated away from the source to the sink, long distance transport, and we just went over that using our mass flow model. Pressure-driven mass flow. All right, the initial short distance pathway of solutes is probably symplastic. They actually thought that the symplastic route was the only route that solutes took, but now they know that, and this depends also on the plant species in question, that the sugars might move entirely through the symplast, or they might enter the apoplast prior to loading in the phloem. So obviously, even when something's been shown to be correct for many, many years, it should still be investigated, because you never know when something new will be discovered. In other words, yes, let's keep reinventing the wheel. Absolutely. All right, so here we have just the symplastic pathway, symplastic loading of sugars into the phloem. So here we have our mesophyll cell, photosynthetic cell, sugars are synthesized, and they will move short distance via the short distance pathway through the plasmodesmata. So this is the symplastic pathway. Sugars are also synthesized in this cell. Movement, sugars synthesized here, and now we're in a bundle sheath cell in this particular example, still following the plasmodesmata route into the companion cell and then loading into the sieve element. So 100% symplastic loading. All right, let's look at the difference here with apoplastic loading. So we start using the symplastic route through the plasmodesmata from cell to cell, short distance pathway, and now, so sugars are moving through the plasmodesmata here, but they are also moving through the apoplast. here, and you can see here also crossing the apoplast to enter the companion cell, and then from the companion cell, once again, through the symplastic route into the sieve element. All right, how does this happen? How do sugars move through the apoplastic route? Of course, there is a transporter involved, and this particular transporter that's been studied is the sucrose proton symporter, which is thought to mediate transport of sucrose from the apoplast into the sieve elements. Oh, someone's dropped out. Oh, you're back, Luke. Your Wi-Fi's holding up okay. All right, so let's have a look at this image down here. And we start with our proton ATPase, which will actively pump protons from this side to this side. And of course, this requires energy from ATP, hence the name proton ATPase. This sets up the gradient, proton gradient, across the, actually this is not across the membrane. Oh, it is across the membrane, yes. Across the membrane, but the proton gradient is actually set up between the, across the plasma membrane into the apoplast. So this region here simply represents the apoplast. And you remember that the apoplast is that region in between the plasma membrane and the cell wall. Okay, so that makes sense. That's where the proton gradient's being set up across there. And of course, the protons will move back down their gradient, and they do so through the sucrose proton symporter. The movement of protons back down their concentration gradient, down their electrochemical gradient, will drive the movement of sucrose across the apoplast. And we're nearly there. All right, now let's look at phloem unloading. Import into sinks, such as developing roots, tubers, and reproductive structures. Involves 3 steps. Phloem unloading, of course, the process by which imported sugars leave the sieve elements of sink tissues. Then once again, we have short distance transport. So after unloading, the sugars are transported to sink cells by means of a short distance pathway. So of course, this is just the reverse of phloem loading. And then finally, storage or metabolism. Oh, that's okay. No worries, Lachlan. I'll see you in a couple of weeks. All right, not a problem at all. And you're all good with your assignment that's due in 2 weeks? Okay, talk to you then. All right, step 3. Let's say yes, I like that. All right, step 3, storage. So the sugars are either stored or metabolized by, of course, mitochondria in those sink cells to provide energy. And of course, all those cells in the root need a constant supply of sugars so that they can generate their own ATP via the mitochondria. All right, and we're going to finish by going over how photosynthetases are actually distributed within the plant. In other words, allocation and partitioning, 2 terms that are often used interchangeably, but they just mean slightly different things. So allocation, the term allocation refers to regulation of the distribution of fixed carbon, in other words, sugars, into various metabolic pathways. So we can say that allocation occurs at a cellular level, allocation of sugars within cells to various pathways, whereas the overall distribution of photosynthates within the plant itself is termed partitioning. So I think just a different scale that we're looking at there when we talk about allocation and partitioning. Allocation includes storage, utilisation and transport. So let's look at what carbon that's been fixed in a source cell can be used for. In other words, what are the carbohydrates used for? Number one, they can be used to synthesize storage compounds, and the number one storage compounds in plants is starch. Starch is synthesized and stored within the chloroplasts. So the carbon's not going very far in this case. And then, of course, the starch is mobilized for translocation during the night. Number two, metabolic utilization. So this is where carbohydrates are used within various cell compartments of the cell to meet energy needs. And of course, the number one compartment would be the mitochondria. Carbohydrates being used in cell respiration, or the carbohydrates may provide carbon skeletons for synthesis of lots of other compounds in the cell. And we'll leave that one there. Number three, carbohydrates may be used as transport compounds. That is, they're incorporated into transport sugars, and we looked at a few different types earlier on for export to various sink tissues. And these can also be temporarily stored in the vacuole of a cell. All right, so that was allocation. Now let's look at partitioning. So as we were actually discussing before, sinks will compete for exported photosynthate. And this competition can determine the distribution of those transport sugars among the various sink tissues of the plant. And there's lots of different ways of actually testing what determines sink strength and how important different sources are to those various sinks. Translocation to sink tissues depends on the position of the sink in relation to the source. So that's what I was describing earlier with that labelling of the leaf with carbon-14 and then looking at where the products end up. Also, young leaves might compete as sinks with roots for photosynthates. Competition's been shown by numerous experiments in which removal of a sink tissue results in increased translocation to alternative and hence competing sinks. Conversely, increased sink size, for example, increased fruit load, decreases translocation to other sinks, especially the roots. I think an interesting demonstration that we used to do years ago, and this would apply equally to grape bunches, I'd say. So this is wheat, and here we go. Our two flag leaves. And what we would do, so this is the developing head of grain or ear, depending on who you talk to. And what we would do, we would cut half of that. So that would be, I suppose, similar to thinning a bunch of grapes, for example, or thinning out fruit on a fruit tree. And what you would expect if you halved the actual size of the ear, you would expect this half that's remaining to become twice as large or twice as heavy in terms of grain yield or grain size in comparison to an intact ear. That's what you would expect, but it actually doesn't happen. Even though it does, the grains actually increase in size and they are heavier. If you just look at individual grains, you certainly don't get a doubling of in the weight, in the total weight. So how do you explain that? How do we explain what's going on there? Any idea?
No, not really. We did a similar thing this year with bunches.
Oh, yes, yeah.
We had some very heavy crops.
Right.
So in some Chardonnay, we actually cut every second bunch off.
Yes. Same sort of concept, yes.
Same sort of concept. I mean, we wouldn't expect them to get much heavier than they already were. It was more from a quality point of view to see if we could see a difference in quality. In the end resulting wine. You know, in a couple of months down the track, we'll do a trial and see whether we can pick up a difference in quality as far as the crop sort of Chardonnay versus the normal crop sort of Chardonnay. So yeah, it's interesting to see what happens when you do halve that crop.
Yes, yeah, I think it's very interesting because obviously there's been a redistribution of photosynthates that would normally go to this strong sink. There's been a redistribution, or another theory is that these leaves that are generally the two most important leaves in providing photosynthates actually lower their photosynthetic rate. Because the sink strength is not as strong as it was.
The sink doesn't need as much.
Yeah. So they either reduce photosynthetic rate, or because the sink is not exerting such a great pull on the photosynthetes are redirected elsewhere in the plant. They're being redirected to different tillers, because of course wheat plants develop these additional stems, which we call tillers. So maybe the photosynthetes are being redirected there. Maybe they're being redirected to the root system, which I doubt, because when a plant's entered that reproductive phase, it doesn't normally redistribute additional resources into the root system.
Yeah.
Because the priority seems to be the reproductive structures.
With grapevines, if you trim the grapevines during the vegetative season, then they shoot laterals.
Yes.
But if you do it later, once the fruit hits a certain size, it's usually after pea size or into veraison.
Yeah.
Then it won't shoot as many laterals. It'll focus all of its energy into ripening the fruit.
Yeah, that's right. And I've seen I'll just get rid of this so I can show you. I've seen a mutant plant. Where is it? Clear. A mutant plant that was completely sterile. It couldn't produce any seeds. It was completely sterile. And this plant, eventually, let's just say that's an inflorescence. Sterile, couldn't produce any seed, sterile, and so on. What this plant eventually did, let's say it normally grows to about this height, it just kept going and going and going, and it didn't stop. But what it did, it was producing all these sterile inflorescences for weeks and weeks after it would normally have started senescing. But not only that, it stopped producing leaves. It actually put all its energy into trying to reproduce. It was quite amazing.
Yeah, that's interesting.
Yeah, really interesting. And it just kept going. I mean, it looked more and more unhealthy as it kept sort of growing and desperately trying to reproduce. But yeah, it was amazing to see that priority into going into the reproductive structures in the plant, even at the expense of leaf production. So obviously it was using only the stems as photosynthetic material.
Yeah.
Yeah, and not even worrying about leaves at all.
And it'd be interesting to look at what nutrient levels it's using in different times, like, you know, how as during flowering, plants will use different
That's right. Yes.
During fruit production and vegetative production, they're using different. Yeah, that would have been interesting to see.
Yeah, absolutely.
Just what nutrients it was using at that point.
Yeah, absolutely. And I think we've got a plant nutrition lecture coming up. Actually, we've got, after tonight's lecture, we have lecture 10, which is carbohydrates. Then we've got 11, 12 and 13. So only 4 lectures to go.
Yep, okay.
Yeah. Carbohydrates, plant growth, plant nutrition, and then plant signalling.
Okay.
Yeah, so not much more to go.
That's good.
Yeah.
It's interesting stuff for sure.
Oh, it is. It really is. And of course, this is, you know, this is just, you know, the fundamentals. There's so much more out there, so much information out there. Now, before you go, I'd just like to remind everyone that the first assessment is coming up, or the first assessment due date is coming up, which is assessment to the Plant Physiological Management Plan. This is a 1,200 word assessment worth 30%, and it is due in 2 weeks' time. Okay, so if you haven't started, I'm hoping that you've started, that you've all started on this. If you haven't, this is certainly a good one to get started as soon as possible. So due in 2 weeks, 3rd of May, and I'll just point out to you, I'm sure everyone's read the subject outline, but just to remind you that there's a fair bit of information on this Plant Physiological Management Plan. If you go to the appendix of the subject outline, here we go. So from here, we go Plant Physiological Management Plan. So you've got a fair bit of information written here for you. And then even better, go to Appendix B and read Table 2. A guide to considering management inputs, physiological processes and details to optimise your management decisions. So this will tell you, give you some management inputs that you might consider, for example, irrigation or rain-fed system, and then the physiological process that you should be discussing, and then of course any management decisions based on the process or the input. So you've got quite a few examples here regarding what you should be talking about, or what you could be talking about, sorry, because you can't always come up with a management strategy for every plant physiological process. So we've got nutrients, insecticide application, herbicide application, pruning, cutting, crop thinning if appropriate, obviously very appropriate for grapevines, fruit trees and so on. Soil conditioning, so you can consider soil pH and so on. And here we have the marking criteria for this assessment. So also have a look at this. This also gives you really important tips as to what you should be including in your assessment. Okay, and a really important part of this that I'll just point out to you. Here we go. Make sure that you talk about the physiological process and link that to your management process. So don't just talk about the actual management input, talk about the science of it as well. In other words, tell Nikki about photosynthesis. Tell her about carbohydrate allocation and partitioning and how that relates to your management input. Okay, so as I said, if you haven't started yet, please read this information. It's really helpful and it'll give you lots of ideas about what to include in the assessment. Okay, Luke, how are you going?
Good.
Good.
Yeah, thank you very much.
No, you're welcome. Thank you for zooming in.
That's all right.
And enjoy the rest of your time in Adelaide.
Yeah, no worries. Are you around around Epping next week?
Yes, I'll be around.
All right, I might try and catch up with you at some point.
Yeah, not a problem. If I'm not in my office, most likely I'm teaching.
Yeah.
Which of course always on a Monday and Tuesday afternoon I'll be teaching. But apart from that, I should be around.
Okay, I'll shoot you an email once I work out when I'll be up.
All right, not a problem. I'll catch up with you then.
No worries. Thanks.
All right. Thank you, Luke. All right. Bye-bye. Yeah, you have a good one too.